Hello Beaver Science fans. We are back today with a video summary of the cell cycle and the phases of mitosis. This summary will include basically a drawing, a hand-done drawing of a diagram that represents the cell cycle that can be found in Chapter 9 of your textbooks, Biology in Focus. And so in the second edition, this is figure 9.6, and this is a cell cycle. This is the first drawing that we'll do. And then the second drawing will consist of the phases of mitosis, which are kind of laid out in figure 9.7 for you. And if you take a look at this, we'll be doing this drawing basically vertically, whereas these are laid out horizontally. And we'll look at each one of the phases of mitosis. We'll start with interphase and then prophase. We'll kind of skip this pro-metaphase and jump straight to metaphase, anaphase, and then telophase to wrap these drawings up. So that's where you can find that information in your textbook. There are some diagrams in Canvas for you to help you with this. And so under Unit 4 in Canvas, there are some handouts. They're labeled cell cycle handout and cell division diagrams. And so the first one kind of looks like this, basically the same diagram that you find in your textbook. And then here are these series of basically the phases of mitosis. So it runs through interphase, prophase, metaphase, anaphase, and then finally telophase. Along with these diagrams, there is a kind of a hand-drawn or a computer-generated graphic of the cell cycle that includes a little bit more detailed information than is included in the diagram in the textbook. And then on the back side of that, what we'll be doing today is basically an overview of mitosis. And here's an incomplete illustration of where we'll start with our drawing today for mitosis. The second video will be a summary over meiosis in the male, spermatogenesis, and then the last one is meiosis in the female, which is oogenesis. So essentially all you need to get started is a blank sheet of typing paper and a pencil or pen to write with, and let's see if we can't get this ball rolling. Okay, at the top of your page, let's label this the cell cycle. And because it is a cycle, essentially that means it's kind of a continuous sequence of events. And basically this is, as a cell goes through cell division, it will kind of follow this general series or sequence of events. And so the cell cycle is made up of 4 phases or stages, if you will. And again, in your textbook, this is essentially figure 9.6. And we'll use pretty much all of the paper. We'll kind of keep the top half of the paper basically will be used for the cell cycle itself, and then we'll do a little bit of labeling kind of down on the bottom. So we start off, this is going to kind of look like a wagon wheel. And so the first phase or stage of the cell cycle is what's called G1. And G1, your book will explain it as a gap phase. To me, essentially, it's kind of like almost like a growth phase. And so during G1, the cell is actually preparing to divide. And keep in mind kind of what we're doing here. If we're looking at mitosis, essentially we're taking one cell and we're going to divide it. We're going to make that one cell into 2. And so in order to be able to do that, you know, again, kind of keep it simple in terms of understanding what's going on in this larger picture of things. But in order to go from one cell to two cells, there are some things that have to happen. Okay, one of the things that needs to happen is we have to increase the number of organelles and the volume of cytoplasm so that both of the daughter cells can get all of the cytoplasm and nutrients and organelles that they need to function. So that happens during G1. During G1, we're going to increase the number of organelles and the volume of the cytoplasm. And remember some of those organelles that we talked about. The energy organelles, the mitochondria, if you're talking about plants, chloroplast, and ribosomes for protein synthesis. And so all of those organelles will need to increase the number of those and also the volume of the cytoplasm. That happens during the G1 phase or stage. The next phase or stage of the cell cycle is the S stage. And this is when we get the brains of the operation. So during the S stage, DNA is going to replicate. And you know a little bit about DNA replication from our previous unit on DNA, RNA structure and protein synthesis and DNA replication. That's all happening in this S stage of the cell cycle. And so, for example, in the nucleus of your cells, you have 46 chromosomes. Each one of those chromosomes will replicate during this S stage, and for a short period of time, you'll have 92 chromosomes in that cell before those 92 chromosomes are divided between the two daughter cells. And if we're talking about mitosis, then we get back to that original chromosome count of 46. And then the next stage or phase of mitosis is G2. And G2 is really final preparation for cell division. So any loose ends, anything that needs to be taken care of, that happens during the G2 stage of the cell cycle. So this is final prep for cell division. Proteins, checking to make sure that the DNA is replicated accurately, kind of just the last thing before we go into the actual process of dividing the cell. And these three stages, G1, S, and G2, those three stages together are called interphase. And that takes about 90% of the time in terms of cell division. So the first three stages, G1, S, and G2, all called interphase, takes about 90% of the time as we go through this process of cell division. So the vast majority of the time is actually spent in preparation for cell division, not actually dividing the cell. I'm going to bring this little loop around here and see if I can make this a little more of a circle for you. And then the last phase of cell division, or the cell cycle, is the M phase, and that's called the mitotic phase. And so this is the M phase. Now if you take a look at this cell cycle, you have G1, S, and G2, and then the M phase. So we have a total of 4 phases or stages to the cell cycle. This is called the mitotic phase. And it takes about 10% of the time. This is when we're actually going to divide the cell. And so when you hear mitosis, mitosis is really occurring in the M phase. This is where we're going to divide the nucleus and then divide the cytoplasm. And so all of this stuff up here, G1, S, and G2, that's all kind of preparation just for cell division. So now we're actually prepared, we're getting ready, we're going to move into the process of cell division. And essentially there's two types of cell division, and depending on the cell, it will go down one of two pathways. It will either go through mitosis or a special type of cell division in sexually reproducing organisms called meiosis. Mitosis happens in what we call the somatic cells. These are your normal body cells. And so that's like your skin cells, your muscle cells, your nerve cells, your blood cells. Those are your somatic cells. They go through mitosis. And we say that the chromosome count is preserved. And so under this type of cell division, we would go from 2N to 2N, or diploid to diploid. Meiosis is a kind of a special type of cell division that gives rise to the gametes. And in meiosis, we're going to reduce the chromosome count, and so we would say we would go from diploid to a haploid chromosome count. The gametes in the male are the sperm, and in the female are the eggs. This is occurring in the reproductive organs, and so meiosis in the male is occurring in the testes, and meiosis in the female is occurring in the ovaries. If we break this M phase down, and you may have seen these terms before. So we have interphase, which is kind of the first three stages of the cell cycle. And then if we break the M phase down, that can be broken down into prophase, metaphase, anaphase, and telophase. You might see pro-metaphase mentioned in the book from time to time. This is kind of the more traditional, to me, what are really the 5 phases of mitosis: interphase, prophase, metaphase, anaphase, and telophase. But don't let that confuse you if you see that extra phase kind of thrown in there in the textbook. But essentially, this is the cell cycle, and it's just a generalized kind of process that the cell goes through as it's going through cell replication or cell division. Go ahead and move on to taking a closer look at what's happening during each one of these phases of mitosis. And so to do that, I'm going to be using this kind of blank sheet. Again, this is available to you through Canvas. Here is kind of that cell cycle, maybe a little bit cleaner, and see if we can zoom in on it a little bit maybe and see the details of it. But you can find a copy of this, again, under Unit 4 in Canvas for you. And then on the back side of this, this is where we will actually go through and draw up each one of those phases and talk a little bit about what's going on. This might be a little bit longer video, so we might be pushing 20 minutes on this, so kind of just hang in there. But if you need to back up or pause or take a look at something, you should be able to do that. So this is our mitosis overview. And in mitosis, one of the things to remember is this chromosome count, what's happening with the chromosome count. So in mitosis, we say that we go from diploid to diploid. 2N is just a kind of a shorthand version of writing basically the term diploid. And diploid, the di means 2, and so diploid means that you have 2 copies basically of the same kind of chromosome, one from your mother and one from your father. In the nucleus of your cells, 46 chromosomes, but occurring in 23 pairs. Here we're going to try to keep it a little bit simpler and just work with a total of 4 chromosomes. And so this is our beginning cell, or our parent cell. And this cell is, we're going to say, is starting out in interphase. And this cell is going to be diploid. Di means 2. And so this cell would be 2N. Here we're only going to have a total chromosome count of 4, and so this would be a chromosome that would have come from the father, and then a chromosome that would have come from the mother, and then another chromosome here, and then its pair. And so this is a diploid parent cell, 2N, and that 2N number equals 4. So that tells you that you have 1, 2, 3, 4 total chromosomes. 2N tells you that those chromosomes are occurring in pairs. And so this chromosome would be carrying the same type of genetic information that this chromosome would be carrying. For example, maybe hair color or eye color. So they will be essentially alternate forms of the genes on those pairs of chromosomes. Okay, this is our DNA. And the DNA early on, before it's kind of replicated and condensed to form those duplicate chromosomes, it's in this loose, unconsolidated form called chromatin. And so again, interphase. Remember that DNA is going to replicate. And DNA replicates basically during interphase of the cell cycle, but specifically during the S stage of the cell cycle. So this chromosome will replicate. It comes down here. It's held together by this little constricted region called the centromere. This chromosome replicates, comes down here. And the same for these 2 chromosomes down here. Okay, this happens by late interphase. In animal cells, you will also see the formation of the centriola pairs. The centriola pairs basically are structures that will be associated with spindle fibers later that will help in kind of providing a mechanism for the duplicate chromosomes to separate. And so this would represent late interphase. And these are our duplicate chromosomes. By late interphase, that DNA has started to go from the chromatin form to this more kind of condensed, tightly coiled chromosome, or duplicate chromosome form. These duplicate chromosomes will also be called sister chromatid. They're held together by this little constricted region called a centromere. These, only found in animal cells, not plant cells, are called centriola pairs. And you may see some additional terms thrown out there, centrosomes and some other terms. There's a lot of very similar terms kind of associated with these processes, and so kind of keep track of which one of those structures relates to the different features of the cell. This little constricted region that holds the duplicate chromosomes together, that's called a centromere. Alrighty, so let's see if we can pick this up a little bit and get to the end before we run out of time. The next cell is, so after interphase we have prophase. And during prophase, the nuclear membrane starts to break down. And so the chromosomes, the duplicate chromosomes, have now completely kind of condensed and formed those pairs, those sister chromatid, still held together by that little structure, that centromere. The centriola pairs will start to kind of move towards opposite poles now, and you'll see the formation of these spindle fibers, which are these protein strands that will eventually attach to the centromeres and kind of force those duplicate chromosomes through the middle of the equator of the cell. So these are our centriola pairs with the associated spindle fibers. And then the little dotted line represents the nuclear membrane breaking down. And this is all happening during prophase. The breaking down of the nuclear membrane, that really is the beginning of the first major process of cell division, which is called karyokinesis. And so in prophase, karyokinesis begins. And it really begins with that breaking down of the nuclear membrane. The next phase is metaphase. And during metaphase, basically, the duplicate chromosomes are going to be, or sister chromatid, are going to be forced to line up through the middle or the equator of the cell. And that's happened because the centriola pairs have kind of migrated to opposite poles. And the associated spindle fibers have attached to those centromeres, and it kind of pushes or moves those duplicate chromosomes to the middle of the cell. So these are our spindle fibers attached to the centromeres. Okay, the nuclear membrane is completely gone at this point. The next phase of mitosis is anaphase. And during anaphase, this is the first phase of mitosis where we see the duplicate chromosomes pull apart or separate. And so they're going to start to move towards opposite poles of the cell. And they're going to essentially follow these spindle fibers back. And so those spindle fibers will still be attached to the centrosomes. And a little piece of that centrosome will stay attached to each what are now the individual chromosomes as they move towards opposite poles. And so during interphase, the duplicate chromosomes have separated. And then the last phase is telophase. And this kind of really represents more late telophase. Because you can see now that the two daughter cells are kind of completely separated. This wouldn't happen until the very end of telophase. And what happens is basically one of each of the pair of duplicate chromosomes will move towards each of the two daughter cells. The dotted line represents the reforming nuclear membrane. And so this chromosome from this pair would come to this cell. This chromosome from this pair would come to this cell. And the same for these two. And you got that little constricted region that stays with what are now the individual chromosomes. A centriola pair stays with each of the reforming daughter nuclei. And so this is our nuclear membrane starting to reform. Both daughter cells, if you'll take a look at what's going on with the chromosome count, both daughter cells look a whole lot like the parent cell. And so both daughter cells are diploid. And essentially, if you compare them to the parent cell, you have 1, 2, 3, 4 copies of the chromosomes that are occurring in these diploid pairs. So I'll try to kind of maybe bring it to the top here and then let you see the overall diagram, and then we'll kind of maybe focus in just a little bit and zoom in and let you see the individual phases of mitosis. And so we start with interphase, and DNA is going to replicate during the S stage. By late interphase, we have the duplicate chromosomes or sister chromatid held together by the centromere. In animal cells, you have the centriola pairs forming. From interphase, we move into prophase. The nuclear membrane starts to break down. You get the formation of the spindle fibers associated with the centriola pairs. They're going to attach to the centromere. By metaphase, the nuclear membrane is completely gone. The spindle fibers have attached to the centromeres. Centriola pairs move towards opposite poles, kind of forces the duplicate chromosomes or sister chromatid to the middle or the equator of the cell. And then in anaphase, the duplicate chromosomes begin to separate or pull apart, move towards opposite poles. And by telophase, the nucleus or daughter nuclei start to reform for each of the two daughter cells. And by late interphase, we have completed karyokinesis and cytokinesis. And these two daughter cells essentially will return back to, remember it's a cycle, back to the beginning, back to interphase. And so this will be available to you, both the handouts and the incompleted form, and then there is also a PDF that shows a completed version of these handouts on Canvas for you. Thanks for joining.